So we're excited to start off with great talks today. And so I'm going to pass it over to Celia Martinez-Jimenez, and she can start her presentation. Thanks, Celia.
Hi, hello. Thank you very much for the invitation and the opportunity to be here today. Today I will be chairing the session, and I'm going to be the first speaker. So I think I will start briefly introducing myself and then the slides. So
actually, may I add one more thing before you begin? I'm sorry.
Yes. Sorry.
Let everybody know in the audience to when you have questions to use. There's a Q&A button down at the bottom of your screen. Please use that to enter your questions in. We'll address them at the end of each talk. And then we'll also have a roundtable session towards the end of the day, and where we'll have some open questions. So please use that rather than the chat. The chat is not monitored by all presenters, so please use the Q&A. Thank you. Sorry about that.
Nothing. Perfect. Thank you very much for helping me with that. Let me see if now you can see my slides.
Not yet.
Not yet. Yes, wait a second. Share screen. Maybe now.
Now I see your screen. Perfect.
Perfect. Good. Thank you. So yes, then please, as Mason was mentioning, just ask your questions in the Q and answer button below. So my name is Celia Martinez, and we are based in Munich, in Germany, and my group investigates the impact of cellular heterogeneity on tissue function. We are interested on the common molecular mechanisms between aging and chronic diseases, and we pay particular attention to the liver as a tissue model. So in the liver, there are several cell types that they crosstalk to keep liver homeostasis. We have the Kupffer cells, the lymphocytes, the endothelial cells, stellar cells, and hepatocytes. During aging, what we observe is that there is some of the hallmarks of aging. They are related with the immune system. As we heard yesterday during the talks, there is a change in the inflammatory pathways. But today, well, I will focus my talks are in two additional hallmarks of aging in the liver. One of them is the lipid droplet accumulation, and the second one is the increase in ploidy or polyploidization. So then during the talk, I will be mentioning these three main topics. I will talk about cellular heterogeneity, mentioning changes in the cell type or changes in the cell type composition. I will talk about a non-deleterious perturbation, and what we have done is to create transgenic mice that they are haploinsufficient, and only one copy of the gene has been activated, and those mice that have been aged over time. And the third concept I will talk today is about polyploidization. Polyploidization is a characteristic feature of hepatocytes, and until now it has not been dissected at the cellular level in the single cell atlases, and has a controversial role. In some cases has been described as a beneficial effect, and in other cases has been associated to senescence and loss of cellular function. So ploidy is a condition of possessing more than two complete genomes. In mice, it's here summarized that polyploidization starts after weaning. At around day 22, start increasing the level of polyploid cells and decreasing the number of 2N cells. So in a nutshell, we have that in an early development, the majority of the cells are 2N, and in a young adult, about 3 months, 50% of the cells are 2N and 50% are polyploid cells. And these tetraploid cells, they can be mono and binucleated. But polyploidization is not only a feature of liver. It has been also shown in skeletal muscle and heart. There are multinucleated myofibers and cardiomyocytes. In the lactating mammary gland, there are also binucleated epithelial cells in the placenta and in the bone, as in the megakaryocytes and the osteoclasts. But recently, it has drawn the attention that maybe polyploidy or ploidy could be a potential clinical target. And there are, this is a summary of 2 different groups that they have manipulated ploidy in the context of an induced liver model. So the first one, with a RAS-induced liver tumor model, this Diril et al., they increased the level of ploidy, and they found that those livers, they had less tumors. And on the other hand, the work shown by Wilkinson et al., they use a then-induced liver model, and when they manipulated with a manipulation and downregulation of a target gene, they decrease the ploidy level. They found more tumors. So in that case, was having a harmful effect. So in this context, we wanted to investigate what is the role of cellular heterogeneity during aging. For that, we have used a methodology that we established recently in the lab. We call it single nucleus RNA-seq2. And it's based on the SMART-seq and foot length transcriptome. The advantage is that has been tailored to work with frozen archive samples. So this is the summary of workflow. We dissect the tissue and then we flash frozen. Then from frozen archive tissues, we can isolate nuclei. We can sort them by ploidy, 2N, 4N, 8N, and so on. And then we miniaturize the reactions in liquid handling robots and perform as an improved version of the SMART-seq2 approach. Just in a nutshell, that method relies on a more efficient lysis buffer. So we can increase the number of genes that we detect per nuclei, about 40%. And that gives us a good number of genes, about 40,000 genes, compared with other approaches, whether they are nuclei or cells. And that was important for us because, as I will show you, we are interested in transcription factors and transcription factor target genes. And with the single nuclei approach, we were able to dissect those factors. Another of the topics that we also mentioned yesterday during the session is the choice of sorting cells or sorting nuclei to dissect this cellular heterogeneity. In our case, we are interested in ploidy, right? And then if we use a dye, and in this case host, and then we do FAC sorting, then we can dissect between diploid and tetraploid. But what we have seen, and this has been reported in the bibliography, is that the 4N mononucleated, those cells that have been related with bad prognosis and poor prognosis in human cellular carcinoma. There has been an increase in non-alcoholic fatty liver disease and during aging. And in these mononucleated tetraploids has been also described processes of aneuploidy and chromosome desegregation. So that was also one of the reasons that we wanted to focus on single nuclei to dissect 2N and 4N hepatocytes. So then with that approach, we studied aging. We started studying the wild type, the C57 black 6 mouse. We isolate the nuclei and perform single nucleus RNA-seq2. This work has been led by Kelvin from my lab and Maren from the Theis lab. Maren now is just a group leader at Calico, and this is a work unpublished that's now prepared for submission. So I hope you find it interesting and leads to many interesting questions. So with the single nucleus RNA-seq2, first we detect all the major cell types in the liver. We have hepatocytes, hepatobiliary cells, endothelial cells, Kupffer, lymphocyte, and stellate cells. We can now have the resolution and see that the majority of the tetraploid cells, they are clustering or they are associated to the hepatocyte clusters, as we were expecting. When we focus on aging, we can see that in this case, as it has been shown in other tissues, we don't find a subcluster associated with old cells. But we did find changes in the cell type composition in the aged liver with an increase in this inflammation and immunoregulatory processes. Our mice, they were all healthy. They had a healthy phenotype, including the classic accumulation of lipid droplets with age. We can see in the H&E the accumulation of fat in the old, and when we stain with DAPI and beta-catenin, we also observed increase in ploidy with age. And in the bottom panel, you can see the increase in the quantification of this lipid accumulation in the old, the decrease in 2N, and the increase in high levels of ploidy. With this approach, we can also study the other cell types, the non-parenchymal cells. And we can observe how there is a downregulation of specific marker genes for those cells. And we can also study the cellular crosstalk about cell types. We studied the transcriptional variability during aging, and we observed that in the wild type, there is a general increase in transcriptional variability in the majority, almost in all the cell types, but we don't find an increase in variability in the hepatocytes, not even when we were able to dissect between 2N and 4N hepatocytes. So we focus on that, on the 2N and the 4N hepatocytes, and we studied the genes that then they were differentially dysregulated during aging. And we found that it was interesting that in the 2N, we found a larger proportions of genes that they were downregulated. So here you have these scatter plots. These are genes, and these are on the right panel, the genes that are downregulated during aging. And we can find that there are more genes in the 2N than in the 4N hepatocytes. Another thing that we were investigating is the transcriptional regulatory networks. What we did is to study the co-expression patterns between the upstream regulators, like these two illustrative transcription factors, CPP alpha and HNF4 alpha, and this co-expression pattern with the downstream target genes. And we found that during aging, the 4N hepatocytes, they also maintain the complexity of the transcriptional regulatory networks. And this, it is consistent for the 4N and the 2N hepatocytes. So now I have shown you that we have used single nuclei to characterize the single cell level heterogeneity in the hepatocytes during healthy aging. But then we were interested in studying what happened if we have a perturbation and what happened during aging with this perturbation. So what we did is to generate two haploinsufficient mice for these two key upstream transcriptional regulators, CBP alpha and HNF4. In the full body knockout of these mice, they are lethal. They are either embryonically lethal or neonatal lethal. And then what we did is to have only one copy of the gene deleted. Those animals are viable, and then we can study the effect of aging. And now we have the ability to monitor how this deletion happened in the 2N hepatocytes, because then only one copy of the gene is deleted, or in the 4N hepatocytes, where we will have, at least in theory, 2 copies of wild types and 2 copies of the knockout allele. So these mice are showing a healthy phenotype. With the H&E, we can see that they have an accumulation of fat during aging. There are no major signs of fibrosis that we monitor with Sirius red, and neither apoptosis. We don't find an increase in apoptosis of these models. When we quantify the lipids, it was very interesting to observe that these haploinsufficient mice, they showed a mild steatosis or a mild accumulation of livers during aging. And then we interrogated and quantified the levels of ploidy. In the old, we observed an increase in ploidy with age. That's something we were expecting. But what we did not expect is to observe an increase in ploidy in the young, only in the haploinsufficient mice. So both mice, the HNF4 and CBP alpha, showed a decrease in the 2N and an increase in the 4N and 8N hepatocytes. Then we further characterized the downstream target genes of these genes that were deleted. And I have just depicted here some of illustrative examples of these downstream target genes. What I call this is like a complex phenotype, in the sense that now that we can distinguish between 2N and 4N, we find differences in the young, differences in ploidy, as well as differences by age. What I mean is that, for instance, in ACOS1, that is one of the genes that involve in lipid metabolism, we can observe that in the young, there are differences in expression between 2N and 4N, and that difference is also maintained in the old. In other cases, in the case of the cytochrome 2b1, we can see that there is a downregulation during aging, and that also is different between 2N and 4N. And a similar pattern is also observed for CBP alpha and their downstream target genes. So we also investigated here the transcriptional variability, how this deletion of one of the alleles was affecting the overall transcription. And in this case, we did observe an increase in transcriptional variability in the 4N hepatocytes, suggesting that there are other processes and regulatory processes participating at the transcriptional level. So until now, I have shown you that after a perturbation, a genomic perturbation, when we have a deletion of an upstream liver-specific transcription factor, we observed an increase in early tetraploidization, so ploidy increases in the young individuals. And when that happened, there is a buffering effect with upregulation of genes that they are involved during aging. And the result is that we have mild fatty liver, we have a healthier phenotype. So we were wondering whether this early polyploidization is something that is only associated to liver-specific regulators, or it is a mechanism that could be more general to cope with a genomic perturbation. So for that, we generated a third haploinsufficient mice. In this case, we use CTCF haploinsufficient mice to monitor that perturbation. Similarly, the full knockout leads to embryonically lethal phenotypes, but the haploinsufficient mice is viable during aging. So CTCF is a structural transcription factor, has many roles as an insulator and as a transcription factor. But even when we delete one copy of this regulator, we also observe an early increase in tetraploidy, polyploidization in the young. So for this factor that is not liver specific, we also observe an increase in ploidy in the young individuals. We also observed that the phenotype of the old mice, they saw a mild fatty liver during aging compared with the wild type, similarly to the other liver-specific knockouts. What I found interesting is that when we did single nucleus RNA-seq2, we did not observe subclusters according with the strain or with the genotype. So all the hepatocytes are clustered together. And without previous knowledge or prior knowledge about the ploidy content, all the hepatocytes, they are very similar to each other. So then what we did was to aggregate all these single nuclei transcriptome in a pseudobulk, and then we compare what are the common signatures. So we found only 4 genes that they were commonly downregulating during aging in all the strains, and those 4 genes that were related with normal development. What I thought that would be more prominent is the signature during aging. I thought that all the strains, they will share a aging signature. And it was surprising for me that there were only 2 genes commonly downregulated across all the species. And these 2 genes that were involved with the lipid metabolism. Sorry. So when we focus on age-related marker genes, for instance, SERPIN3K, that has been shown that to be downregulating during aging, in we check the pseudobulk, we can observe a downregulation in the wild type and a downregulation in all the strains. When we then can dissect the role of 2N and 4N in young and old, we can observe that in the 4N of the haploinsufficient mice, there is an upregulation of this gene, and this upregulation is also maintained in the old. For the cytochrome 2b1, this gene has been characterized as one of the genes that is upregulated during aging. And then in the wild type, when we study the pseudobulk, we can observe the upregulation. And then we can see that there is a differential transcriptomic program between 2N and 4N in the young and in the old. So let me show you just a snapshot of these genes. And that is the take-home message I would like to transfer with these results and all these violin plots. That, for instance, the SERPIN3, and I have shown you, is downregulated during aging. And we can see that the 2N and the 4N, they are downregulated in the old. But when we have an haploinsufficient mouse that also shown a global downregulation of the gene, we can now observe differential profiles between 2N and 4N. And the 4N in the young, they have the ability to buffer or to rescue this phenotype of aging. So then we wanted to investigate why this is happening, or how we can model this mechanism. And then we thought that if the 2N, they replicate and they have a faithful mitosis, then the daughter cells, they should be wild type and KO. And similarly for the 4N. But there is a random mitosis, there is a larger repertoire of genetic configurations that could be out of this outcome. One of the possibilities and the hypothesis we had behind of this study is that maybe the 4N and this have this buffering effect because they can rescue the phenotype and somehow they can bring or non-randomly select the wild type allele. To check that hypothesis, we compare the transcriptome between the wild type and the knockout. Sorry. And in the 2N, we observed this transcriptional difference that we were expecting because it's an haploinsufficient mouse. However, in the 4N, we found that they are very similar. The profile of the 4N in wild type and the haploinsufficient mice, they are very, very highly correlated. And the same happened in the young and the same happened in the old. Interestingly, this is a feature that we observed in all the knockouts and during aging for HNF4, CBP alpha, and CTCF for liver-specific and non-liver-specific transcription factors. Then our next question was to find out what is the mechanism, how we can validate this non-random selection of the wild-type allele. So for that, we use a base scope. And this is summarized in this sketch. So we designed a probe in green that was binding the exon 4 and 5 of the HNF4 locus. And then we have a different probe in red that was binding to the excise allele configuration after CRE. So it is binding to the 3 and the 6. In that way, just monitor the presence of green and red dots on the cells. We can monitor whether we have a wild type genotype in 2N or in 4N. We have an haploinsufficient configuration, one allele wild type and one allele knockout, or even the full knockout configuration. And with that, we examine in the young and in the old haploinsufficient mice the presence of these genotypes, and then we quantify them. So it was very interesting to see and surprising to see that in the old, we found an increase the presence of the wild type cells with the wild type allele. And that is what prompted us to say or to suggest that the polyploidy hepatocytes protects during aging with the non-random selection of the wild type allele. So with that, I want to finish and wrap up with this take-home message. And I have shown you that aging leads to an increase in hepatocyte ploidy and regulatory complexity in both in 2N and 4N hepatocytes. Also that the haploinsufficiency of tissue-specific master regulators increase metabolic buffering via this early tetraploidization. We have also seen that this increase in ploidy leads to a beneficial profile in liver and non-liver specific haploinsufficient mice, and that the hepatocyte ploidy protects maybe through this non-random selection of the wild type allele. And then just to leave also some thoughts for the audience and perhaps for discussion. These are the possible impacts of this finding is that maybe cellular heterogeneity is not only associated to a detrimental characteristic. It might be beneficial depending on the tissue or the age. Also, we might need to reconsider senescence in the liver and take it into account this concealed heterogeneity at the level of ploidy in a homogeneous population of hepatocytes. And I believe that ploidy could be really a therapeutic target for treating chronic liver diseases. So I would like to thank Kelvin, Mateos, and Yanis are one of the main drivers in the lab, and all the members of my lab for their input and feedback. Maren, a computational partner on the project, from Fabian Zeiss, and now at present is at the group leader at Calico. Duncan Odom, and of course, all our facilities that they support our work. And thank you very much, and I'm happy to take questions now.
So Vera, I see you have your hand up. So if you want to ask the first question.
Thank you. Well, Celia, fascinating, absolutely fascinating. So I have two questions. So first one, do you think this selection happens just because cells have survival advantage or like proliferative advantage, or there is really selection on the level of mitosis, like somehow the cell knows where the bad chromosome is?
Yeah, thank you. Thank you very much. We don't observe an increase in division, cell division. So we find when we do our cell cycle analysis, we see that the majority of the cells are in G1. So I think that the most likely scenario is that somehow they sense and they can select positively for this wild type allele. So these cells, they live long in the liver. So maybe just in the first round of replication that they have that ability to select. So this selection has been shown in other liver models of more acute damage. But in this case, we don't have an increase on apoptosis. So I think it's a mild phenotype, but still it helps the cell to decide what to keep.
And then my second question, kind of a crazy thought. So like why other cells don't benefit from the same strategy? Because you can imagine like in some plants, for example, wheat, it's all like polyploid and they just become more robust. But in mammals, I guess polyploidy would somehow be detrimental for other tissues. So what happens to transcriptional networks, for example, in other tissue? What would polyploidy disrupt?
So at the organismal level, yes, that is too complex to handle. Like it can only happen in plants, in some reptiles. But the heart is also polyploid. And there are some reports showing that after heart infarction, there is also an increase in polyploidization. So the precise mechanism how the cell is regulating several copies, I think is still not clear. But there are evidences showing that other tissues that they are also polyploid, they might use this system to protect the tissue.
Thank you.
Thank you.
Celia.
Hi, hello.
Here, I'm Dou. So this is a very interesting talk. I have two questions. The first question is because you brought up senescence. So I wonder whether you see any difference for your 2N, 4N, or even 8N cells during aging, right? So whether you see senescent cells are preferentially enriched in particular N. Yeah,
very good question. We check the genes of senescence and the healthy aging, and we didn't see an increase. But it's true that there is a limitation, that is single nuclease RNA-seq. So for that, we know that some of the classic markers of P21 and P16, they are not captured with the single nuclei. So that I have not detected them doesn't mean that they are not there. But with the single nuclear resolution, we cannot find them. So some of the classic gene signature of senescence, we don't find them with a single nuclei RNA-seq. So I'm not sure whether they are not at all, but at least I can tell you that we don't detect them. Yeah, thank you.
So it could be is irrelevant. So another question I have is that have you looked at the cell fusion? And so I know the hepatocyte, a lot of them have the cytokinesis failure. The other thing that can change the chromosome number is the cell fusion and sometimes even the nuclear fusion. So do you think this plays a role during aging in hepatocyte?
So there are several mechanisms for polyploidy in the liver, and one is the ones that you have mentioned. We have not checked. With the resolution we have with the nuclei, we have intact single nuclei. So it's something that would be interesting to go back to the histology and check. The limitation is that since we have a snapshot that we cannot monitor in a long period of time, I don't know if there will be cause or consequence of the aging process.
By the way, have you ever seen an odd number nuclei in the hepatocyte, like 3 and 5? Have you ever seen anything like that?
With FACS sorting, we have not seen it. So we can only see complete duplication of the genome, like 2N, 4N, 16N. So to study that, we will need to go to other kind of approaches at the cellular level to study aneuploidy. Thank you very much.
Gotcha. Thank you.
Peter, I can see your hand.
Yeah, hi Celia. Really wonderful talk. Thanks. So you've highlighted the benefits of this the kind of the buffering through ploidy, but there would also seem to be potentially a lot of kind of regulatory detrimental effects through overexpression of certain genes. So I'm just wondering how do you think the cell copes with the negative consequences of this? Do you have any insight into that?
So in this particular case, we see that it's beneficial because we have a phenotype. We have a better, healthier phenotype in all the haploinsufficient mice. What we have seen in the young is that polyploid cells, they also adjust their transcriptional levels according to their zonation in the liver. So when we studied the ploidy 2N and 4N in the young, and we did a single nuclei and RNA scope, we found that the 4N that is close to a position in the hepatic lobe adjusts the expression level of the genes according to its location. How does that regulation? I don't know, but there is a kind of plasticity, and maybe this is what we can see with this transcriptional variability in the 4N. There are other mechanisms that they are playing a role when you have more copies.
Okay,
and it can be depending of the insult or depending of the age that could be beneficial or detrimental.
Okay, yeah, that's amazing stuff. Thank you.
Thank you.
So we have quite a few questions also in the Q&A section, and I'd like to remind everyone to please use that. It's at the bottom of your screen to post your questions. So if you like, Celia, I can sort of pose some to you. Okay. So the first one's from Margarita Mir. Looks like two-part, and it's: does tetraploidization happen in human as early in life as in mice? And then the follow-up is also, does it go beyond 4N? I'm not sure what the 4N is.
Yes, thank you. Yes, in human also happens early in young individuals, but there are no reports of a precise timeline on at what age they start. So also increases with age, but the numbers of high levels of ploidy, they are not as high as in mice. For instance, in the mouse liver, it's about 80% of the hepatocytes are going to be polyploid, and in human, in an adult human, it's about 50–60%, so it's a bit lower. And yes, there are also higher levels of ploidy. You can find 8N, 16N as well in on the livers, mainly with age. So in the young animals, you will find 2N, 4N, 8N, but if you want to find 16N, then you have to move to old individuals. Thank you.
So the next one is, do you have to consider semi-dominant recessive phenotypes which might not be reflected simply by transcription levels?
So Yeah, that's very interesting. I don't know if I have evidence to answer that. We have started the characterization mainly focused on the downstream target genes that are metabolic genes for hepatocytes. But these haploinsufficient mice, they are whole body haploinsufficient. So actually it could be other phenotypes that could be affected. We have focused on that because those transcription factors, they are liver specific. And then we started the analysis on those downstream liver genes.
Adriel Hernando says, thank you for a really interesting presentation. He has two questions. How does polyploid hepatocytes, or how are they able to differentiate the wild type allele from the mutated allele for aging protection? And then the second question is, how does the 3D genome affect cell polyploidy since CTCF is related to the genome architecture?
Thank you. There are some hypotheses for that. So there are some work in human cells where they also see a non-random selection of alleles. One hypothesis could be because of the ratio of proteins that are bound to the chromosome are different. And then there is a kind of sensing machinery. The other possibility is homologous recombination, that then it could select the wild type by recombination. We don't know, we have not explored how CTCF affect the genome architecture, but we know that the haploinsufficient mouse CTCF has minor effects in the transcriptome. This has been already previously published by Duncan Odom's group. But it's something that, yes, CTCF has other effects that are not only as a transcription factor.
So next question is, are very long-lived humans or other species showing an increased relative ploidy in comparison to normal life expectancy?
Hi, thank you, and hi, Robert. We don't know. We are now investigating that for other long-living species. And I think the key point will be to be able to have a longitudinal cohort where actually we can see several time points and not only young and old. So we are working with some other species, but I don't have the results yet. Thank you.
The next one is from Roxandra Lambuta. She says, fantastic talk. I was wondering whether you tested the selection of wild type allele can be either accelerated or detrimentally affected by ploidy reduction and repolyploidization cycles driven by liver injury.
Thank you. We have not tested that. I believe that yes, depending on the injury, if there is an acceleration of the cell cycle, then those ploidy reduction and polyploidization might have a prominent role. In this specific haploinsufficient mice, they have a healthy phenotype. There is no selection for the viable hepatocytes, so it's not the main. It's not the main factor in the ploidy induction.
So I see somebody posted into the chat, Salvatore Olivero. So I'm going to ask that one. He says he has a question about liver transplantation, as liver is the only organ that can be transplanted from very old donors. But this increase in ploidia, could that be detrimental?
So there are no, I have not seen any report on transplantation in human. What I can say is that with organoids, it has been shown by Meritzel Hatch group that they can generate organoids from 2N and 4N hepatocytes. So both of them, they are able to regenerate and repopulate and have the ability to maintain the stem cell properties and regenerate. The good thing of the 4N hepatocytes, and this also has been reported, is that if they need to divide, they have to reverse to 2N. And that is why they are beneficial as a tumor protector, because if there are more foreign cells to generate a tumor, they have to go back to N and then duplicate. So in the context of transplantation, I could speculate that the 4N would be kind of a safer option than the 2N, because it will need more time to go back to 2N and then duplicate. Thank you.
So this question is does not have who posed it, but are there differences in the total levels of transcription in the polyploid cells?
Total level of transcription, meaning total number of genes transcribed per cell. We observed about 1.2 higher number of transcripts. The cells, the nuclei are slightly bigger, but that overall increase is not, it doesn't affect the changes that we see in specific genes. So what we observe is that even with this 1.2 increase in transcription, we find genes that are upregulated with ploidy, downregulated with ploidy, genes that they don't change the mean expression of ploidy, but they change the gene expression distribution. So all the possibilities they are also happening within ploidy.
So Celia, our next speaker doesn't seem to be logged in yet. So if you're okay with it, we can spend a little extra time answering all these questions.
Yeah,
if that works for
happy to
you.
do so.
Great. Yeah, there's still a few more. So One Ronda says in mice, the hepatic polyploidization occurs just after weaning. Is this linked to nutritional complexity, switching from breast milk to chow? And they say thank you for an interesting talk.
Thank you. It has been shown that it's triggered by glucose, or by insulin signaling. So in hepatocytes in vitro, manipulated the level of glucose and the insulin signaling, you can promote polyploidization. So it is thought that the change of diet and that metabolic stimuli is what is triggering the polyploidization in the liver.
Payal, it looks like you have your hand raised. Feel free to go ahead and ask your question. Good morning.
Hi.
Oh,
we cannot hear you.
Yeah, you muted yourself, Payal. Sorry.
Sorry. Yeah, I was just saying that, yeah, that was a great talk. Thank you. So I was just wondering, like, the increase of polyploidy with age, does the DNA level and the histone dosage compensate, or do we expect to see that with age, with more DNA, maybe the histone dosage doesn't quite scale, and that might lead to some problems?
That is a very interesting question. We don't know yet. Not knowing 2N and 4N with that level of dissection, we don't know how the histone or the histone modifications, they might have a different role there. I don't know. I think it's a very good question. Thank you.
So I see, it looks like Gary has a question, I think probably from our group in Carlsbad. Gary, do you want to open your mic, or do you want me to just ask it? I guess I'll just go ahead and ask.
Please just go ahead.
Got it. All right. So his question from our group in Carlsbad is, has ploidy ever been seen in brain tissue?
Not that I recall. I don't remember any.
Simple question. Okay. Harris asks, do you see any change in senescence by immunocytochemistry?
We are trying to verify that actually that we don't detect senescence in the single nuclei is actually similar in the tissue. So we need to validate that our level of detection we can get the right conclusion. So we have not checked yet in the tissue. Thank you.
Jessica Prouse asks, HNF4A overexpression is increasingly being considered as a therapeutic intervention against NFOL than HCC? How would you predict HNF4A or CBPA overexpression to influence these polyploidy phenotypes or vice versa?
I don't know. I don't know, because what I'm thinking is that having an overexpressed transcription factor means that now the ratio between transcription factor and binding site is going to change. So intuitively, I believe would be beneficial, just because to have a upstream regulator might enhance the lipid metabolism and help on the differentiation of the hepatocytes. But I'm not sure whether the ratio between the proteins and the binding sites, they might have other kind of secondary effects. This I will think about this. Thank you. And I see Peter.
Hi, Celia. So liver cancer tends to is more common in older people than younger people. So I'm just wondering, what's the impact of polyploidation on tumor genesis? I mean, do liver cancers tend to have an unusually polyploid genome, or is there some selection perhaps for the the less polyploid genomes, which might be they might actually have a selective advantage in terms of their outgrowth and clonal expansion? So I guess I'm just generally wondering what you think the implications of this is for tumor genesis in an old liver.
So it has been shown that human cellular carcinoma has more polyploid cells, and they have also more mononucleated cells. What I'm wondering is whether that comes as a consequence of an insult.
Yeah.
So we have only the snapshot, and now with these young insufficient mice, now I'm thinking that we should revisit some of those samples, taking into account the age, and maybe in mice we can model this tumor progression and to see whether the damage starts early or later and together or not with polyploidization. So if these polyploidy cells, they are beneficial, it could be a consequence of a damage. The polyploid cells, they increase to rescue the wild type or the healthy allele. But then if this cannot happen because there are many cells affected, then the final outcome will be a polyploidy tissue. So I'm not sure yet. For that, we will need longitudinal studies where actually we can pinpoint component.
Yeah. Okay. Thanks.